We were talking about the large intestines in the last one, and I was trying to give you kind of a acclimate you to what you're looking at. So the large intestines surround the small intestines more or less, and you have the interface area. It's the ileum and the ascending portion or the ascending colon of the large intestines. That's where they connect, and they connect via this valve, the ileothecal valve, and you actually have a pouch there, which we call the cecum, that can collect. So when the substance is secreted from the ileum into the cecum, kind of collect there until um, I guess alert, maybe do the final alert to the large intestines that something is coming in. And so what happens is, again, we have all the layers of the GI tract. So you have the mucosa, the submucosa, the muscularis externa, and you have the serosa on the outside. And so you have these muscles that actually can move this along into the different segments, so up, against gravity in the ascending colon. Another area, if you follow the ascending colon up, that's on your left, the area where it turns and becomes the transverse colon, we call that the hepatic flexure. We call it that because that's more or less where the liver is. Transverse colon, and then a descending colon. You also have here, you have what's known as haustra. Those are those little, it looks like little globs of material stuck together. And then you have a band that kind of holds the whole thing together, and that's labeled the I don't see the label. Oh yeah, I do. The 10A coli. 10A coli. That's kind of the band that's running all the way through the whole thing. And you can see this. These tend to be things that are asked. I don't guarantee I'll ask it, but tend to be asked. Anatomy that gets asked. Inside, you can see the inside is very different than the small intestines. You do have some ripples, but there's minimal absorption taking place here, mostly water, and no digestion taking place. There's some vitamins that are generated because of the bacteria in the large intestines. And then it ends with the rectum into the anal canal. Now we'll look at that in closer detail because we want to look at what the defecation reflex is like. Here's the rectum in even closer detail. We're not looking at the defecation reflex yet, but just note that you have two sphincters. So you have an internal sphincter and an external sphincter. And you can see the internal sphincter is actually listed underneath the external. So you have the external first, external because it's the furthest on the outside of the actual tube, which makes up the GI tract. Internal, inside. The internal sphincter is involuntary. The external sphincter is voluntary. They're both muscular structures, close and opening, and it's just a matter of whether it's voluntary, involuntary. Involuntary is going to be under your autonomic nervous system. Voluntary is going to have, you have skeletal muscle voluntary control of it. And then lax villi, but lots and lots of goblet cells. So there's no villi in it, but lots of goblet cells because there's a need to have lubrication. And as a matter of fact, that image is pretty good, but look at this image. Look at how many, all of those little, what look like little air bubbles in the purple portion, those are all goblet cells. Those are all cells that are able to secrete mucus. And that's really how much you need in terms of the mucus in the large intestines. Wall, very muscular. One of the reasons I brought this one in, I do kind of like microscopic slides, I can tell you that. But one of the reasons why I brought this in, it's a good review with regard to those layers, outermost, serosa, muscularis, or muscularis, that's the muscularis externa, submucosa, and then mucosa. And then you have a little bit of the thing that what we are used to. It's not really villi, but you do have a almost a organization with regard to the portion that's closest to the lumen. And then the defecation reflex. It looks complicated. It really isn't that complicated. What you have is you have the presence of compact matter in the lower portion of the colon. So that compact matter is going to cause stretch receptors to be activated. So you have step one there, and what you're doing here is the blue is going to be your short reflexes, and remember, short or intrinsic are ones that are just in that particular area of the body. Long reflexes are the ones that are communicating with your CNS, your whole systemic working of your nervous system. So you have stuff, fecal matter, stretching areas in wherever it's existing, so stretch reflexes. That's going to stimulate the muscles for one thing, but it's also going to stimulate the parasympathetics via the sacral portion of the parasympathetics. It's going to stimulate the somatic motor neurons. So the somatic motor neurons are going to think about your skeletal muscle neurons, that neuromuscular junction of the skeletal muscles. That then increases the contraction of those muscles, so it's going to increase peristalsis throughout the entire large intestines. Remember, this is central nervous system, so that stimulation is going to make everything in the large intestines move, all of the muscles, and that will then in turn cause there to be a distension, so more space, and that fecal matter will be able to move on down, and that causes more stretch, move on down, more stretch, until it gets into the position where it's actually pushing against the internal sphincter. The internal sphincter is going to relax, and then the external sphincter is going to be pressured. This is when you have that urge to go, and then if in fact you have the opportunity, you will have a voluntary reaction of that external sphincter, and then everything will move out. It's a great process. One that the only thing that we really control is the external sphincter. That's the only thing that is a learned behavior, I guess you might say. Defecation reflex goes back to short reflexes, long reflexes, and also the acting on the muscular layer of the alimentary canal. Switching topics a little bit, I guess you might say, we're all done with the anatomy. Now we want to look in a little bit more detail, or review even kind of some of it, is how do these nutrients get absorbed? What are the processes? Almost all of it happens in the small intestines. About 80% of electrolytes in almost all food. The rest of the electrolytes are going to be absorbed between the large intestines and maybe a little bit in the stomach, not much, but the large intestines. Most are absorbed by active transport. That means we use ATP to digest our food, active transport. Some are absorbed in a passive way in terms of transport, meaning transport proteins, but most are absorbed with ATP use. So glucose, amino acids, nucleic acids are absorbed by secondary active transport with sodium. Tends to be a trading one to the other. Iron and calcium require transport proteins. These are some of the reasons why we eat vitamins and minerals, because they're cofactors for taking other things in. Sodium, Na+, is a ion, so anions follow sodium. Remember, or learn, I guess, A, D, E, K are the fat-soluble vitamins. They're carried in as a large fat globule. So they're carried in by bile salts, so large fat globule, absorbed in, they're carried into the, as much as they can into the bloodstream, but also to the lymphatic system. But remember, the lymphatic system dumps into the bloodstream via the inferior vena cava. Much of vitamin K is absorbed in the colon. That's one of the big functions of the colon of the large intestines is vitamin K. Water-soluble vitamins diffuse with the exception of B12. B12 needs the intrinsic factor that is made way back in the beginning of the whole process, the intrinsic factor for B12. And B12 is important for making red blood cells. I do have some other slides for this. This is just where it's written out, but I have actually two. This one, again, might work better for you, where you have glucose, secondary active transport with sodium, and there was the word I kept forgetting, monosaccharides. And then fructose is facilitated diffusion. Remember, facilitated diffusion is actually passive as long as there's something to facilitate it. Amino acids, active transport, dipeptides and tripeptides transport with hydrogen ions, and short chain fatty acids. These are the only fatty acids that are absorbed straight into the bloodstream, the short chained. And then the long chained, they are going to be taken into the lacteal of the, it's a lacteal, that's what that structure is. I don't know if I've used that term, that's found in the villus of the small intestines. That's what we associate with the lymphatic system. They're taken in what's known as a chylomicron, which is the fat globule surrounded by bile salts. And what you're seeing on the left is the apical surface of the epithelial cell, which is the brush border, the microvilli, and then what you're seeing on the right is the interface between the epithelial cell, sorry, kind of tongue-tied, and the blood capillary. I do have another one coming after this. It might help you too with this. This one, just to review, this is the villi, and this is looking at that lacteal. The green portion is the lacteal, the lymphatic vessel. This is how large lipid molecules are absorbed, as well as those fat-soluble vitamins are absorbed this way. You also have, I guess, the expression of the short-chain fatty acids and the monosaccharides, the amino acids, absorbed. And then another image, another way to look at it with a little bit more information about what's happening in other parts of the body with carbohydrates. And I think you have a spotlight in your book that's really good for this too. The spotlight, it's named Chemical Events of Digestion, which is really nice too, with colors, etc. I just couldn't get it to come up clear on the PowerPoint. Monosaccharides are absorbed via co-transport with sodium ions, except for fructose, which passes facilitated diffusion. They enter the capillary and then transported to the liver. So all of this most of this is transported to the liver. You also see there the enzyme sources needed to actually break those disaccharides into monosaccharides. It's actually when it gets to the brush border that you become a monosaccharide. You can see the brush border enzymes that are going to actually turn the disaccharides into a monosaccharide. All this happening in the mouth and small intestines. Protein digestion, transported co-transport with sodium, and then to the liver. You have pepsin turning protein into large peptides, chains. Then you have pancreatic enzymes, small peptide chains, and then brush border enzymes finishing the job, turning those small peptide chains, so those small chains of protein units, into amino acids, some dipeptides and some tripeptides. All this happening in the small intestines. And we will stop at this one and move on and finish up on the next lecture.